Oops, you guys are not on the right side at all. All right. Oh my goodness. Okay. I apparently just don't. There we go. All right. The recording for this lecture is just going to start with fast forwarding and reversing through the slides. Okay. So on Friday, we did kind of a brief overview of photosynthesis and kind of what it looks like, right? It's split into two main chunks, light-dependent reactions, light-independent reactions. We started to mention, right, kind of how we kind of came to understand photosynthesis. And so now we're gonna, after kind of a broad introduction, get into the actual kind of mechanics of the thing. And first talk about how this light energy is being caught in the first place, right? And that is done by pigments that are found in chlorophyll. So we're going to talk a bit about, well, we'll talk a bit about pigments. Let's talk about pigments. So all a pigment is, right, if you call some molecule or some compound a pigment, it is just a molecule that absorbs light in the visible range, right? We're going to be talking about energy and energy levels and wavelengths and things, but basically everything we're talking about is concerning light in the visible range. That's what plants are kind of collecting and using to do their thing with, right? And so pigments absorb and reflect various wavelengths of light within the visible range. This is what dyes are. They're specific chemicals that reflect specific wavelengths of light and absorb specific wavelengths of light. Yeah. And so the visible color, what you see is just the light that's not absorbed, right? This orangey sweater, right, is absorbing all the light basically between like green and violet, and it's reflecting most of the light between red and yellow is what's happening, right? And so that's kind of the region of wavelengths, the region of light that we're talking about with these pigments. Now, so we've got pigments that are absorbing and reflecting specific wavelengths of light. And so the other end of that equation is obviously the light itself, right? And light is this weird thing. You've been in physics class or just watch whatever, right? The weird thing about light is that it is a wave, an energy wave, and also a particle, right? So it is an energy wave, like a radio wave or a gamma wave or anything like that. But it's also got kind of a physical component to it. It is also a particle. And so it behaves in kind of a weird, funky way. So when we talk about waves, we're talking about the electromagnetic spectrum, which is this very, very broad spectrum, right? With everything from radio waves that are tens of meters long, right, are huge, to things like gamma rays, like radioactive rays that are thousands of nanometers wide, right? But for our purposes, again, we're only looking at the wavelengths that we can see. And so the visible spectrum is specifically right here between 400 and 740 nanometers. So relatively small, but we kind of blow it up. So this is the part we're concerned with, right? So we're specifically talking about this kind of wavelength on the electromagnetic spectrum. And these are where photons are moving, right? This is kind of the chunk we're concerned with. So I already said it, photon, right? So the particle that are moving in these wavelengths are called photons. So they are little discrete kind of packets, particles of energy, right? And the energy of a given photon is inversely proportional to its wavelength. All that means, so you've got a photon, we have waves of light, photons are moving in these waves of light. And so if the wavelength is really short, right, if we're over on that 400 nanometer end, right, the wavelengths are really short. And that means the photon's really high energy. Kind of think of it like vibrating along because those wavelengths are super, super, super short, right? Versus where we get to kind of lower energy light over towards the reds, the wavelengths are longer, right? And so the photon, you can kind of think of it of having this kind of slower, slopey sort of motion, right? So long wavelength, low energy, short wavelength, high energy, right? Little bouncy guys, right? So the amount of energy that photon has, right, determines where it is on the spectrum, whether it's lower energy or higher energy. Right? And the amount of energy the photon has is important because beams of light, i.e. photons, can remove electrons from certain molecules. This is what we're wanting them to do, right? When photons come in and remove these electrons, it creates an electrical current, right? And the whole point of this photosynthetic thing is to harvest the energy from light, right? So this is where we're going to kind of be using these photons. This is called the photoelectric effect. It is the effect whereby light or photons create an electric current. Okay. So light has energy. That energy can remove electrons. It creates an electric current. And so in photosynthesis, the chloroplasts, right, our little double membrane guys, act as photoelectric devices to capture the energy from the photons and use it to do stuff. So they're going to absorb sunlight and then transfer these excited electrons, right? These photons are going to start exciting these electrons, increasing their energy level. And then we're going to take those excited electrons and put them onto electron carriers that we will then use to do stuff. All right. So photons can remove electrons or increase the energy level of an electron. We'll see this. This is what is happening in the light reaction. So photons can increase the energy level of an electron, but only if that photon, turns out the molecules are picky, only if that photon is carrying the right amount of energy. So we've got these specific atoms or molecules, and they're only going to absorb certain kinds of photons, right? Either high energy or low energy or this kind of energy, right? They're only going to absorb photons that are traveling at a certain speed, basically, or with a certain wavelength, right? This is called that molecule's absorption spectrum. What spectrum of photons, what spectrum of light of a photon will increase the energy of an electron on a molecule, all right? So it's just the wavelengths of light that a molecule can absorb most efficiently, right? And then everything else gets reflected. This comes back to that visible light thing, right? The molecules that make up this sweater or that trash can, right, or this table, right? There are certain photons of light that they're absorbing and there's certain ones that they're reflecting, right? This sweater is mostly reflecting lower energy photons for whatever that's worth, right? All right, so the pigments. The pigment usually in question when we're talking about photosynthesis is chlorophyll, right? This is the main photosynthetic pigment in plants. It is what color? It's green, right? This is why plants are green, because they're full of chlorophyll. If you smash a leaf, the green that comes out, right? You're smashing out the chlorophyll, right? And so they only absorb a really narrow range of energy, right? They are absorbing, right? And so this graph, the higher the peak, the more of that light that's being absorbed, right? So if they're green, what are they absorbing if they're green? Yeah. Yeah. Which wavelengths? Which colors? What are they absorbing?
I didn't do good in art class. Yeah, I did not pick this yellow school.
All right, so chlorophyll is green, which means how about this? What is it reflecting? What is it not absorbing? It's not absorbing green. It's not absorbing anything between blue and yellow. Right, you see how low this line is here? So this black line is chlorophyll. And it doesn't absorb anything from blue to yellow. It absorbs anything with red in it, basically.
So if a plant was able to have a larger absorption scale, what would happen to plants? Like if it was able to absorb more varieties. So wavelengths and
Like what would they look like, or what would they?
would it increase the size, or would it increase oxygen producing and CO2 and stuff like that?
Or would they be a different color?
All of the above, possibly, right? Because, and actually they do to some degree. You'll notice there are other lines here. There are other pigments that get involved in photosynthesis.
That's something that they could possibly change the plant. And
Possibly, except for, is that my next bullet? Yeah. So chlorophyll A is the main one. And chlorophyll A is the only pigment that is able to directly convert light energy into chemical energy. So that's where we get a little bit limited, right? So it's chlorophyll A, our very traditional, very green chlorophyll, right, is the only one that can directly do that conversion from photons to energy. Now, plants have accessory pigments. You'll see these kind of other lines, right? Because we've got chlorophyll A doesn't do anything with this light, but we can add some accessory pigments that kind of bring that in and absorb some more, right? A little more into the yellow, a little more into the blue, right? So chlorophyll B is one of those, that's the dotted line. Chlorophyll B still doesn't absorb kind of very specifically green and yellow, but it takes a little bit of red and it takes a little bit more blue light in and kind of increases what a plant can do. And so it complements chlorophyll A by increasing that absorption spectrum. And so chlorophyll B is one accessory pigment. The other accessory pigment that we see are carotenoids, right? So carotenoids are the gray line. And so they actually absorb basically everything from violet all the way up to green. So they have a really wide absorption spectrum here. Which means they're what color? What color are carotenoids? Are they reflecting blue or absorbing blue? So they're reflecting.
Mostly red.
Yeah. Yeah, always think carrots when you see carotenoids, right? They reflect red to yellow. They absorb everything else. So they can absorb a wide range of photons, but they're not really great at transferring energy, which is what happens with these accessory pigments. So chlorophyll A is really great at transferring energy, but it's really picky about what light it will do it with. We can add in some other pigments and kind of take in a bit more energy, but they're not efficient at transferring the energy. So it boosts a bit, but not a whole lot. The other nice thing that carotenoids do is carotenoids are also kind of when we talk about antioxidants, right? Carotenoids are kind of in that category. And so what they do is they remove free radicals from the DNA of the plant, basically damaged DNA. You may have heard free radicals before. So when you get sun damage, like on your skin, yeah. So it is causing free radicals in your DNA. It's damaging your body kind of at the very cellular level, right? And antioxidants, things like carotenoids in plants, are able to kind of mitigate that damage, right? To kind of repair free radicals that occur over time from being exposed to radiation functionally.
Like aloe vera. It's like when they tell you when you get a sunburn to put aloe vera on it to help the healing process.
So that is more of that's not really, yeah, it is a moisturizer, anti-inflammatory. So the part we're concerned about with like sunburn damage is that it hurts. Your skin becomes inflamed from the damage. And so things like aloe vera or even Advil, you can take Advil when you've got a sunburn. Inflammatory response is inflammatory response. Advil fixes all kinds of stuff. Or just wear sunscreen. Step one, just wear sunscreen and try not to like burn your DNA up too quickly.
Yeah.
I mean, it's somatic DNA. It's short-lived, but that's fine. That's for later in the semester. So carotenoids. Carotenoids are also why we get fall color, because chlorophyll A is green. What is picky? So when the days get short, chlorophyll A is like, oh, I'm out of here. They senesce, they go away. The carotenoids hang on for a while, all the pigments that are reds and oranges and yellows, because they're a little less picky. They'll pick up, well, they're less picky about the light they take. So they'll hang on while the days get shorter up to a point. And then of course, eventually we're in the winter, it's too cold, photosynthesis isn't really happening in the leaves so much. Carotenoids senesce, leaves fall off. All right. So that's where you get your fall colors. To create photosystems, just step next, right? So we've got pigments, they're in the chloroplasts. They can capture and transfer energy. And so, yeah. And so they are part of what is called a photosystem inside the chloroplasts. You have these little, we'll talk about them. Boop. All right, so chloroplasts, little green bean guys, full of these folded membranes called thylakoids, right? These little kind of pancake stacks of membranes. Embedded in the thylakoid membrane are these little guys called photosystems, which are filled with those pigments, right? And so photons, right, waves of light are hitting these pigments and they are collecting the energy from these excited electrons and passing them on to electron acceptors, right? Creating these really high energy electrons to transfer onto their NADPs.
Separated into two parts, is that the photosystem?
So the photosystem is separated into two parts and it is embedded in the thylakoid membrane. Yeah. So the two parts, so inside any given photosystem, you've got two things going on. You've got an antenna complex, right, which is receiving the photons, right? The light is hitting the, so if you think of like an antenna or like a receiver, it's collecting the photons, it's collecting the energy. Right, and that energy gets kind of passed through the photosystem until you get to one of these reaction center chlorophylls, and we'll talk more about what a reaction center looks like, right? But that is where the actual electrons get moved around, is in the reaction where electrons are moved happens in the reaction center. So antenna complex, right, are light harvesting kind of pigment molecules. So they're capturing photons from sunlight, pulling that energy into the photosystem. Energy goes into a reaction center, which is also called a transmembrane protein pigment complex. We'll talk about this one, right? It just is what it is, right? So transmembrane, because we're embedded in the membrane, it's a complex of proteins and pigments, and that's it. That's the reaction center. So it's just a term that lists out all the things that are in it, basically. All right, so here's our reaction center. We've got our little chlorophyll, the pigment. We have our electron carrier. We have photon energy coming in, right? So photon energy is coming into this pigment. It's exciting the electron. That's what kind of the height of the electron kind of infers the energy, right? So we've got this little lower energy electron on my chlorophyll, hit it with a bunch of light energy. Electron gets super excited, right? And then we're going to take this high energy electron and put it onto this electron carrier that can then take it onto the electron transport chain. You'll see it later, right? And then this other little guy, he's just an electron donor. He's kind of like, what do you call those things? Like one of those fire bucket squads. So he's just here to grab an electron and refill the chlorophyll, right? So he takes his electron, puts it in the chlorophyll, we hit it with light again, and do the same thing over and over again, right? And he's just like passing electrons into the chlorophyll. Electrons are getting excited, passed on to electron carriers. And so that is the reaction center, just this little kind of collection of pigments and molecules that are exciting electrons and moving them on down the chain. All right. So all of this that we're talking about basically are the light reactions. So we're going to talk about the light reactions, the light dependent reactions a little bit more specifically. So light reactions happen in the thylakoid membrane, which makes sense. It's where all of this stuff is, right? The photosystems are embedded in the thylakoid membrane. And we've got 4 stages. 2 of them we just talked about, right? We're going to have a primary photo event where we capture some light. charge separation where we pull off electrons. And then we've got the last two parts of the light-dependent reactions are things we've already talked about in respiration. We go to the electron transport chain and chemiosmosis. So actually not really that much new here in the light-dependent reactions. Okay. So first three chunks are all part of capturing the energy from the light, right? We're going to collect photons, we're going to move electrons, and we're going to put them into the electron transport chain and make a bunch of ATP, just like we did in respiration, as far as that part goes. All right, so we're going to capture energy from light. Woo! Right, push it through the electron transport chain. Like I said, produce a bunch of ATP and NADPH. So those are the 4 stages of the light-dependent reaction. So we'll kind of look at this, walk through it kind of diagrammed out. This is really cheap. All right, so like we mentioned, photon is captured by pigment, chlorophyll A specifically. It excites an electron, right? That excited energy is transferred to a reaction center where we put the electron on an acceptor molecule like NADP. And that initiates the electron transport chain, which we'll see in a second looks very much like the electron transport chain in respiration, right? It's in the membrane. It's a series of little proteins and carriers, right? Kind of doing a bunch of these little redox, move the electron down the electron transport chain. Doing this moves protons across the membrane, creates a high concentration of protons inside the thylakoid, right? The electrons, once they're done going through the chain, reduce NADPH. And then the proton gradient that we just created moves back across the membrane through ATP synthase and generates a lot of ATP. Okay. It should be looking vaguely familiar, slightly. Now it looks like this. So I feel like, especially with the light dependent reactions, I feel like it's just much tidier than respiration because it's just all right here, right? We've got this folded thylakoid membrane. We got all this stuff stuck in it, all in a nice row. Right, so we've got a photosystem full of pigments, photons come in. So here's this, the line kind of matches electron energy. So we've got photons coming in, we excite this electron and we throw it into the electron transport chain. It hits a little carrier, goes through this proton pump, right? So the moving the electron through pushes protons inside the membrane. Electron gets a little more tired, goes through this carrier, hits another photosystem, we hit it with more photons, energize it again, and pass it on, right? Another carrier, another protein. And eventually that electron goes to NADPH, which will meet us in the Krebs cycle, right? In the meantime, we've been creating this proton gradient. They push their way out through ATP synthase. We make a bunch of ATP. And those are your light dependent reactions. Full stop. So, notice we've got 2 photosystems. Now, one thing to not let mess with you. So the first one is photosystem 2. The second one is photosystem 1. It's just because scientists discovered photosystem 1 first. There's no better reason for it than that. It is what it is. So photosystem 2, we've got this BCF complex as protein. Photosystem 1, NADP reductase. That's it. So, and they work in a series, right? This isn't like the Krebs or Calvin cycle. That's kind of like a circular process. This is just us pushing electrons through all these carriers, right? So it's called non-cyclic photophosphorylation. Non-cyclic photophosphorylation. So photosystems 2 and 1 are linked by an electron transport chain, right? So here's the electron transport chain and carriers that look very much like the ones in respiration, and we just have kind of the photosystem stuck in between, right? The first one is photosystem 2. So it absorbs photons, it oxidizes water, and then passes its electron on to the B6F complex. We pump protons inside the membrane, go on to photosystem 1. And so this electron that we've kind of powered up, used up a bit, we're going to power it up again and pass it on to NADP reductase to make NADPH.
Are you gonna remember the B6, maybe that?
No, probably not. I mean, be aware that there is a proton pump here, but I'm probably not going to ask you to specifically remember B6F. I don't think so. Right, the point is we've got a photosystem, part of the ETC, a photosystem, and the rest of the ETC. That's like the importanter parts. All right, so this is happening. We're changing the energy levels of the electrons kind of repeatedly, right, as they go down through this process. So another way to kind of think about what's happening, we can diagram this kind of just in terms of the energy of the electrons, right? So there's electrons in here attached to these pigments. We bring in this photon energy, it hits one of these reaction centers and we energize this electron. We've got this super high energy electron, we're going to pass it through the carrier, through the complex, through a carrier to photosystem 2, right? And again, we're going to hit it with more photons. And this time, right, because we haven't used it all up yet. So we're going to add kind of the same amount of energy. So we end up with, in this kind of stepwise way, a more excited electron than we had the first time, right? And the same thing, it's going to go through NADP reductase to make NADPH, right? Because if you remember in the last electron transport chain and respiration, we weren't adding any energy. So the electron was just kind of like fading out. We just dropped it on some oxygen at the end because it was just like totally toasted. This time we keep powering it up, right? We keep adding energy. So at the end of it, we've created a proton gradient and we can still use the electron to make NADPH, right? Because we've not totally burned the thing up by the end of it. All right. Right, so that happens. We've made a bunch of NADPH. We've been pumping protons here through the B6F complex inside the membrane. So step last in the light-dependent reactions is chemiosmosis. So we've got all these protons kind of built up here. They want to kind of get out. They're really highly concentrated in here, not highly concentrated out here. They go out through ATP synthase to produce ATP, right? Moving down their concentration gradient. And so every electron pair that gets moved through here yields one NADPH and basically one ATP, a little bit more, basically one ATP. Right, and so that is how the light reactions turn light energy kind of into more usable energy for the cell, right? NADPH and ATP. Right. So light-dependent reactions. So the next step then, if we're creating this energy, the whole point of this is we're able to, if we're a photosynthetic organism, to fix atmospheric carbon. Right, so we've got the light-independent reactions. We've used the light, we've collected the energy, we've stored it in compounds. Now we can use that energy to fix carbon dioxide from the atmosphere, and that's called the Kelvin cycle. Right, so it'll be a cycle. It's going to look similar to the Krebs cycle in some ways. All right, so we're going to talk about carbon fixation, and then talk about the Kelvin cycle, where we're going to convert 6 molecules of CO2 into a glucose molecule. All right. So we're moving out of that thylakoid membrane, right? We've been generating all these NADPHs and ATPs kind of to the outside of the thylakoid. And surrounding those kind of thylakoid stacks is the stroma, that kind of gel stuff that fills the chloroplast. And so all of these products, all this ATP and NADPH is going to power the Kelvin cycle where we convert inorganic carbon into an organic molecule. So similar to the Krebs cycle, it's going to be a series of enzyme catalyzed steps. You will not need to know the enzymes or any of these intermediates. We're just going to talk about it. We're going to kind of break it into 3 main chunks just to kind of talk about what's happening, right? Unlike the Krebs cycle, right? Because the Krebs cycle was breaking down, right? Decarboxylating the last few carbons from glucose. So instead of that, the Calvin cycle is going to be producing new compounds, right? This is one of those kind of like respiration is kind of like photosynthesis backwards and vice versa kind of things, right? So Krebs cycle was breaking down glucose. Calvin cycle is creating glucose. Now, carbon fixation depends on this molecule, ribulose 1,5-biphosphate, Ruby P, or we're just going to call it Ruby P. All right, so Ruby P is going to be kind of like, if you remember, we started the Krebs cycle with oxaloacetate. Ruby P is going to serve a similar function in the Calvin cycle. It's made from some intermediates of glycolysis because remember, plants are creating glucose, but they're also breaking down glucose because they're also, you know, doing their own thing, metabolizing and stuff. So we're going to use some glycolysis intermediates to make a Ruby P, and we're going to use Ruby P to fix carbon dioxide. Now, Ruby P is a 5-carbon compound. We fix CO2 to it. We now have a 6-carbon compound, right? We added a 1 carbon to a 5-carbon. And so it's a 6-carbon intermediate, a C6 intermediate. Just means that we've made a 5-carbon thing and attached a 1-carbon thing to it to make a 6-carbon thing, right? So what happens in the Calvin cycle is we make this 6-carbon thing. We've incorporated, by incorporating atmospheric carbon dioxide, and then we break it in half to make 2 3-carbon things. They're 3-phosphoglycerates. We'll just call them PGAs, right? So we take Ruby P, we attach a CO2, we break it in half to make 2 PGA molecules. We have now incorporated CO2 into an organic molecule. Atmospheric carbon dioxide is now part of an organic molecule. That's it. That carbon is fixed. We did it. This reaction is catalyzed by an enzyme called ribulose biphosphate carboxylase oxygenase, Rubisco. So Ruby P grabs a CO2, Rubisco breaks it in half. Done and done. So that's basically the first phase of the Calvin cycle. So we're going to split the Calvin cycle. A whole lot of stuff is happening. We're basically going to talk about it in 3 chunks or 3 phases. So the first part is generating those 2 PGA, right? So we stick a CO2 onto a Ruby P and we break it in half. We make 2 PGA. Carbon is fixed. Then we're going to reduce PGA. Basically, we're going to change those 2 PGA to 2 G3Ps because 2 G3Ps makes glucose. And we've done it. We've taken CO2 out of the atmosphere and we've made a glucose. Ta-da. The rest of it, right, if you remember, this happened in the Calvin cycle too, right? We only had a couple carbons to break off in the Calvin cycle, and then the rest of it was just making oxaloacetate again. Similar thing in the carbon cycle, right? We fix our CO2, break that molecule apart, zhuzh it around, make a glucose. The whole rest, phase 3, is just making Ruby P again, right? Because it can either, so G3P, if you have a couple of G3P, you can make them into glucose, or that G3P can complete the Calvin cycle and make another Ruby P. And that is it. That is the Calvin cycle, right? It does cost a good bit, right? We're going to burn 12 ATP here, 12 NADPH here, 6 more ATP over here. But that's fine because the light-dependent reactions are running all day. Right? And we're just going to keep converting CO2 into glucose, right? So 6 turns, right? 6 times around creates 2 extra G3P. right? We use 6 CO2 molecules, send them around the Calvin cycle. And so every 6 turns, we get 1 glucose, right? Because we've incorporated 6 CO2s in. We end up with 1 molecule of glucose for every 6 turns of the Calvin cycle. All right, I think this is the last little chunk because, and this leads us into what we'll finish up. There's just one tiny little chunk of chapter 8 left for Wednesday. The first intermediate, right, we make this 6-carbon thing and we break it into 2 3-carbon compounds. Because that first intermediate is a 3-carbon compound, we call it C3 photosynthesis, 3-carbon photosynthesis. All right, there are exceptions to that, which we will talk about on Wednesday when we talk about photorespiration. We talk about C4 and CAM as alternatives to C3 photosynthesis. Yeah, so we're within less than 10 slides from the end of chapter 8. So nearly there.